shigotoxin-producing E. coli are located on the chromosome of bacteriophage as well as on virulent plasmids. Bacteriocins. Many bacteria produce agents that inhibit or kill closely related species or even different strains of the same species. These agents are called bacteriocins. To distinguish them from the antibiotics, which have a wider spectrum of activity, bacteriocins are ribosomally synthesized peptides, although several require extensive post-translational modification for activity. The structural gene for the bacteriocin and the genes encoding proteins involved with processing and transporting the bacteriocin, and for conferring immunity to its action, are carried by the plasmid or a transposon. Bacteriocins are named in accordance with the species or organisms that produces them. The Escherichia coli we have colicins coated by col plasmid. Bacillus subtilis produces subtilism, and so on. The COL plasmid of Escherichia coli encoded various colicins. Colicins released from producing cell bind to specific receptors on the surface of susceptible cells. The receptors for colicins are generally entities whose normal function is to transport some substance, frequency, a growth factor of micronutrient through the outer membrane, the lipopolysaccharide layer of the cell. Colicins kills cells by disrupting some critical cell function. Many colicins form channels in the cell membrane that allows potassium ions and protons to leak out, leading to loss of the cell energy forming ability. However, colicin E2 in the DNA endonuclease that can cleave cellular DNA, and colicin E3 in the nucleus that cuts at a specific site, and the 16S, an rRNA, and inactivates ribosomes. Col plasmids can be either conjugative or non-conjugative. The bacteria or bacteriocin-like agents of gram-positive bacteria are quite different from the colicins, but are also often encoded by plasmids, and some even have commercial value. For instance, the lactic acid bacteria produce the bacteriocin nisin A, which strongly inhibits the growth of a wide range of gram-positive bacteria, and is used as a preservative in the food industry. Engineered plasmids. The technique of genetic engineering discussed later in this chapter have been made possible. The constructive in a laboratory and limited number of new artificial plasmids, incorporation into such plasmids of genes from a wide variety of sources has made possible the transfer of genetic material across the species barrier. It is even possible to synthesize completely new genes and introduce them into plasmids. We now turn our attention to the details of conjugation and show how certain plasmids can act to mobilize the bacteria chromosome, allowing transfer of chromosomal genes from donor to recipient. Bacteria conjugation, mating, is a process of genetic transfer that involves cell-to-cell contact. As we discussed previously, conjugation is a plasmid-encoded mechanism. A conjugation plasmid uses a mechanism to transfer a copy of itself to a new host. However, sometimes other genetic elements can be mobilized during conjugation. These other genetic elements can be other plasmids or the host chromosome. Indeed, conjugation was discovered because the F plasmid of Escherichia coli can mobilize the host chromosome. Mechanism of conjugation transfer may differ depending on the plasmid involved, but most plasmid and gram-negative bacteria seem to employ a mechanism similar to that used by the F plasmid. Conjugation involves a donor cell which contains a particular type of conjugated plasmid and a recipient cell which does not. Because conjugation was discovered by performing genetic crosses, the donors were called males and the recipients were called females. The genes that controlled conjugation are contained in the TRA region of the plasmid. Many genes in the TRA region are involved in mating pair formation, and most of these have to do with the synthesis of the surface structure, the sex pilas. Only donor cells have these pila, explaining why RNA bacteriophages that attach to them are called male-specific. Different conjugative plasmids may have slightly different TRA regions, and the pila may also be different. The F plasmid and its relatives encode F pila. Pila allow specific pairing to take place between the donor cell and the recipient cell. All conjugative and gram-negative bacteria is thought to depend on cell pairing brought about by pila. The pila make specific contact with a receptor on the recipient and then retract. Pulling the two cells together, the contacts between the donor and the recipient cells then become stabilized, probably from fusion of the outer membranes, and the DNA is then transferred from one cell to another. Mechanism of DNA transfer during conjugation. DNA synthesis is necessary for DNA transfer to occur, and the evidence suggests that one of the DNA strands is derived from the donor cell and the other is newly synthesized in the recipient during the transfer process. Some genes in the TRA region have to do with DNA transfer and replication, a mechanism of DNA synthesis in certain bacteriophage called rolling cycle replication is presented. This model best explains DNA transfer during conjugation and possible mechanism for this process is outlined in Figure 10.21. The whole series of events is triggered by cell-to-cell contact, at which time one strand of a plasmid DNA circular is nicked and one parental strand is transferred. The nicking enzyme required to initiate the process, TRA-I, is encoded by the tri-operon of the F plasmid. This protein also has helicase activity and is thus also involved in unwinding the strand to be transformed. As this transfer occurs, DNA synthesis by the rolling circular mechanism replaces the transferred strand in the donor, and complementary DNA strand is also made in the recipient. Therefore, at the end of the process, both donor and recipient processes possesses completely formed plasmids. The plasmid DNA transfer process is highly efficient under appropriate conditions. Virtually every recipient cell that pairs acquires a plasmid. When the plasmid genes can be expressed in the recipient itself becomes a donor and can transfer the plasmid to another recipient. In this fashion, conjugated plasmids can spread rapidly between populations, behaving like infectious agents. The infectious nature of the phenomenon is a major ecological significance because a few plasmid-positive cells introduced into the appropriate population of recipients can, if they contain genes that confer a selective advantage, convert the whole recipient population into a plasmid-bearing population in a short period of time. The widespread occurrence of drug resistance carried by a conjugated plasmid has led to some serious problems for the chemotherapy of infectious disease. However, as mentioned above, plasmids can also be lost from a cell by a process called curing. This could happen spontaneously in natural populations, when there is no selection pressure to maintain the plasmid. For example, plasmids conferring antibiotic resistance can be lost without affecting the cell viability if there are no antibiotics in the cell environment. The formation of high-frequency strains and chromosome mobilization. The F plasmid of Escherichia coli is not only conjugated, but also can mobilize the chromosome for transfer during cell-to-cell contact. The F plasmid is the episome a plasmid that can integrate into the host chromosome. When the F plasmid is integrated into the chromosome, conjugation can lead to transfer of large regions of the host chromosome, and genetic recombination between donor and recipient can then be very extensive. Cells possessing an unintegrated F plasmid are called F+, and those that have a chromosome-integrated F plasmid are called high-frequency of recombination. Both F and high-frequency cells act as donors, but conjugation using high-frequency donor leads to transfer of the host chromosome. Because the plasmid is part of the chromosome, plasmid integration is a very simple mechanism of mobilizing other genetic elements. The term high frequency of recombination refers to genetic recombination between the donor chromosome and that of the recipient. Cells without the F plasmids are F negative and act as recipients. In general, cells that contain a conjugated plasmid are very poor recipients for the same or closely related plasmids. This is because the plasmid is blocked from entering, not because of replicating incompatibility. We thus see the presence of the S plasmid results in the three distinct alterations of the properties of cell. 1. Ability to synthesize F pilots. 2. Mobilization of DNA for transfer to another cell. And 3. Alteration of surface receptors so the cell is no longer able to behave as a recipient in conjugation. The integration of the F plasmid into the host chromosome can occur at several specific sites, called IS for insertion sequences. These sites are regions of homology between chromosome and F plasmid DNA. For a discussion, it shows the integration of the S plasmid involves the insertion of the chromosome at such site. In the particular high frequency shown, the integration site is between the chromosomal genes Pro and Lac. One integrated in the plasmid no longer controls its own replication, but the tra-operand still functions normally and the strain synthesizes pila. When a recipient is encountered, conjugation is triggered just as in an F+ cell and the DNA transfer is initiated. However, since the plasmid is now part of the chromosome after part of the plasmid DNA is transferred, chromosomal genes begin to be transformed. As is the case of the conjugation with just the F plasmid itself, DNA transfers involve replication, so after transfer, the high strain still remains high frequency because it has retained a copy of the transferred genetic material. Thus the chromosome is said to be mobilized. As we mentioned, integration of the plasmid is a simple mechanism of mobilization. Other mechanisms of mobilization are known involving not only the host chromosome but other plasmids. One such mechanism is for a mobilized plasmid to contain an ORIT sequence like that found on the F plasmid. Such a plasmid will be transferred by conjugation if it is in the cell with conjugated plasmids that can supply all the other necessary genes. Since a number of distinct insertion sites are present, a number of distinct high-frequency strains are possible. A given high-frequency strain always donates genes in the same order, beginning with the same position, but high-frequency strains of independent origin transfer genes in different sequences, usually